So, good afternoon, students.
Good afternoon, sir.
So, students, by the time others are joining, if anyone has any question. Okay, so students, let's just see this again. Okay, so this is your MHC class 2 molecule. Okay, it has two chains. It has the alpha chain and it has the beta chain. Okay, there are two domains in each chain, which is alpha 1, alpha 2, and beta 1, beta 2. Okay, and both these chains have a transmembrane segment and a short cytoplasmic tail. Okay, if you see, this is MHC class 1 molecule. It has one chain, the alpha chain. Okay, and there are three segments or domains, alpha 1, alpha 2, and alpha 3, and an associated molecule which is called beta 2 microglobulin. Okay, so only the alpha chain is membrane bound. It has a transmembrane segment and the short cytoplasmic tail. The peptide binding cleft is between alpha 1 and alpha 2, whereas in MHC class 2 molecule, the peptide binding cleft is between alpha 1 and beta 1. So the peptide binding dome in class 1 molecule is alpha 1 alpha 2, and in class 2 molecule it's alpha 1 beta 1. Okay, peptide binding cleft is closed at both the ends. Okay, open at both the ends. It means that this peptide binding cleft is closed, right? So it is closed from all the ends. Okay, whereas in class 2 molecule it is open at both ends, so it is kind of this open. Okay, and that's why it class 1 molecule can accommodate a peptide of 8 to 10 amino acids. Okay, whereas class 2 molecule can accommodate a bigger peptide of 13 to 18 amino acids. Okay, so because it is open, it can accommodate a bigger peptide, right? और अगर ये पूरी तरह से बंद होगा, so whatever peptide comes, it has to fit within it. Okay, so that is, so these are some of the differences between class 1 and class 2 molecules. Okay, and now let's come to this very first picture. So you can see this is MHC class 1 molecule. So they have represented it in this way. We were drawing circles for this. So it is up to you, whatever way you want to present. Okay, but the basic things remain the same. Okay, clear? So I hope you are clear with MHC molecules now. Okay, so now if it is clear, then we can move on to the next topic, which is antigen presentation and processing. Okay. Okay, students. So let's now start with antigen processing and presentation. So students, you can see there are two terms. One is antigen processing. Okay. Another one is antigen presentation. Okay. So students, to make you understand this, we can take a very basic example that you see every day in your life. Okay. So if I ask any one of you, right, how do you make food in the kitchen? Okay. So what will be the basic steps? The first thing is, you know, some food item will, it will come to your house. Okay. Let's just, for example, say potato. Okay. So now just help me what we do with that. Okay. तो सपोज आलू की सब्जी बनानी है, राइट, और तुम्हारे घर के अंदर 4 आलू आ गए. So what will you do then? So who is going to help me? Students, ये तो हम रोज देखते हैं घर में. So you have potatoes. What will you do then in the kitchen?
So,
Yes, Shakti.
yes, sir. We will wash them and then peel them and cut them into pieces, small
Okay.
pieces.
ठीक है. Then what next, Shakti? उसके बाद?
Next, sir, we'll cook it or
Okay.
after washing it, we will put oil on the.
ठीक है. ठीक है.
And
ठीक है. अब बन गया. ठीक है. फाइनली बन गया. उसके बाद?
yes, sir. So we will take it out and serve it.
Yes. Okay. So uh, what she said, let me just try to show it. Okay, I'm just enjoying the way I'm discussing this. Okay, so you have potatoes. Okay, so you don't get such round potatoes, but whiteboard has made it so round. But you got this potato. At this moment, they are outside your house. Okay, now these potatoes you have brought in to your home. Okay, and you have now taken these potatoes to the kitchen. Okay, whereas Shakti said we will wash them. Okay, and then we will peel them, and we will make smaller pieces of this. ठीक है. इसके बाद हमने क्या किया, इसको फ्राई किया. ठीक है, और इसको रेडी कर लिया. ओके, सो नाउ दे आर रेडी. Okay, next she said that now we are going to serve it. Okay, so for serving you'll first take a plate. Okay, or you will take a bowl. Okay, और इसमें क्या करोगे, because you are serving, so you will add right students, clear. मैंने अभी कोई साइंस की बात नहीं की.
Yes, sir.
जो
Yes.
किचन में होता है वही बात की.
Yes, sir. Yes.
अब इसमें थोड़ी सी साइंस लाई जाए.
Yes.
Okay. So ये this is your pathogen. Okay. So this is your pathogen, and this is your body. So this pathogen has entered your body. Okay. Now इसमें क्या होगा? पहले जैसे पोटैटो की प्रोसेसिंग हुई थी. ओके. सो फर्स्ट थिंग वुड बी एंटीजन प्रोसेसिंग. सो ये तुम्हारा एंटीजन है, तो इस एंटीजन की प्रोसेसिंग होगी. ओके. और उसके बाद हमने क्या किया था? उसको सर्व किया इन अ बाउल. ओके. सो दिस बिकम्स योर एंटीजन प्रेजेंटेशन. ओके. ठीक है. Clear? So this is your antigen processing, this is your antigen presentation. थोड़ा सा इसको और ध्यान से देखें. So ये तुम्हारे एंटीजेनिक पेप्टाइड्स हैं. Okay? So these are your peptides and this is your MHC molecule. Clear?
Yes, sir.
This process clear? अगर ये clear है तो तुम्हें एंटीजन प्रोसेसिंग एंड प्रेजेंटेशन का पहला पाथवे क्लियर हो जाएगा. Okay? So now let's come to the slides and understand this process. Okay. So students, antigen processing and presentation अभी समझ आ गया?
Yes, sir.
Basically, we are first processing an antigen and then presenting that antigen in the form of peptides to the immune cells. Okay. So these are two different things, antigen processing and presentation. Okay. Now students, again there are two things. If you remember, we talked about different types of antigens, right? So जब हमने पहली क्लास में एंटीजन की बात की थी तो मैंने बताया था कि देयर आर एंडोजेनस एंटीजेंस जो कि बॉडी के अंदर होते हैं. ओके. एंड देयर आर एक्सोजेनस एंटीजेंस, एंटीजेंस व्हिच कम फ्रॉम आउटसाइड द बॉडी, right? So endogenous antigens are synthesized within the cells, okay, including self and unself proteins, okay, and they are presented through class 1 MHC molecule, right? So body के अंदर for example normal cellular proteins होते हैं, tumor proteins होते हैं, viral and bacterial proteins होते हैं, right? So all these kind of proteins are generated within our cells, right? So they are endogenous antigens, right? So they are formed within us, but still they are, antigenic, right? Because they have developed later and our immune system has not seen them, right? So these are your endogenous antigens, and then there are a lot of antigens which every time keep coming inside our body from outside. For example, a bacteria, virus, fungi, anything which is coming from outside. Okay. So these are processed through MHC class 2 molecules, right? Okay, so we have already seen these two processes. One is antigen processing. So this is basically conversion of native proteins to peptide. Okay, and why this has to be done? Which can combine with MHC molecules, right? सो MHC मॉलिक्यूल का स्ट्रक्चर तुम देख चुके हो. जो MHC क्लास 1 मॉलिक्यूल है उसमें 8 से 10 अमीनो एसिड का पेप्टाइड फिट होगा, राइट. क्लास 2 में 13 से 18 या 9 से 18 की रेंज का पेप्टाइड फिट होगा. ओके, so the whole bacteria will not come and fit into this MHC molecule. So the bacteria needs to be processed, broken down into smaller peptides so that those peptides can fit into these MHC molecules. And this MHC molecule जो कि हमने डायग्राम में as a bowl दिखाया था, this can take up the peptide and then go out and show it to the immune cells. Okay, so that is antigen processing. Other thing is antigen presentation. So this is a display of peptide MHC complex on the surface of antigen presenting cells, and obviously when you are going to present it to someone, or when you are going to serve to other people, only then other person can eat. ठीक है, वो बात अलग है कि वो किचन में आके खा जाए इज अ डिफरेंट थिंग, बट कोई गेस्ट आया है सपोज, you will take it outside and then it will be seen by the guest. Okay, so similarly here, this MHC peptide complex, it is formed inside the cell and then it moves out, it is expressed on the cell, and then it is then recognized by the T cells, right? And you now know which T cells recognize which MHC molecule. So for MHC class 1 molecule, which T cell? Students? Yes, cytotoxic T cells. Okay. So I hope these things are clear: endogenous antigen, exogenous antigen, antigen processing, and antigen presentation. Okay. Students, there is one more thing. When we talk about B cells, okay, so B cells have these membrane bound antibodies through which they will bind to the antigen, okay, and then there will be proliferation of these B cells, and there will be production of a lot of antibodies, and these antibodies will go and bind more antigen, right? So this is how your B cell work, right? But T cell, they cannot recognize antigen directly like the B cell. Okay, so T cell have to be activated. Okay, so if T cell sees a bacteria, right, there is no direct proliferation or anything. So T cell does not directly get activated. Okay, so the T cells, they do not recognize native antigens, right? Therefore, the native antigen has to be broken down into smaller peptide. Therefore, T cells have to be, the antigen has to be processed so that the T cells can recognize that antigen. Is this clear? Okay. So now students, we have antigen presenting cells. और कौन-कौन से हैं? स्टूडेंट्स, एंटीजन प्रेजेंटिंग सेल्स कौन-कौन से हैं? पिछली क्लास में बताया था.
Macrophages, dendritic cells, and B cells.
Yes, macrophages, dendritic cells, and your B cells. Okay, so they are your antigen presenting cells. In antigen presenting cells के अंदर, okay, the antigen will be processed, and these hands, they are like your MHC class 2 molecule, okay, and they will present the antigenic peptide to T cell, and if it is MHC class 2 molecule, then it is presented to T helper cells. Okay. Okay, so students, just to explain this concept, just to show that T cell activation, it occurs because antigen is processed and presented. Okay. For this, some experimental conditions were set up. Okay. Now, you take an antigen presenting cell. Okay, it could be any cell, and then you fix it. And what do you mean by fix it? That now it is fixed, it cannot do any activity. Okay, every function is ceased, slows. Okay, it is just fixed, it cannot do anything. Okay, so in one of the experimental condition, antigen presenting cell लिया, उसको fix कर दिया, and then incubated with the whole antigen for 1 hour. Okay, and after that T cell activation was checked, and there was no T cell activation. Okay, in another condition, antigen presenting cell was taken, it was incubated with the whole antigen, okay, for 1 hour, and after 1 hour it was fixed. Okay, and in that case T cell activation was observed. Okay, in another case, antigen presenting cell was taken, it was fixed and then incubated with antigen peptides, not the whole antigen but only the peptides, and then it was observed that T helper cells got activated. So ये समझ आया? कोई समझा सकता है ऐसा क्यों हुआ? Anyone? Okay, so students, just yes, anyone?
Sir, would it be fine if I tried?
Yes, go ahead.
So I think in the first case, there was no T cell activation because there wasn't enough time for the antigen to be fully processed by the antigen presenting cells. Whereas in the second case, the antigen is incubated for an entire hour with the antigen processing cells, and only then it is fixed. So after that, that I'm guessing was enough time for the antigen to be processed, and so the T cells were activated. And as for the third case, it's not the whole antigen that was added, it was only the antigen peptide. So it's like the processed antigen was already there, so that also made T cell
Okay.
activation.
Okay. So Manaswini, you basically said that in the first case, there was less time, but you can see here, it was incubated for 1 hour, the same time which was given here. Okay, but the problem here is that the cell is fixed, and when we say that the cell is fixed, it means it is not doing any activity. Okay, so in this case, because the cell is fixed, it is not carrying out the antigen processing. Okay, and when antigen processing does not happen, the peptides are not formed and it is not loaded, right? So until and unless this peptide comes here, the T cells will not recognize it. Okay, in this case, before fixing, antigen has been added, right? So now in this 1 hour, the antigen is broken down into smaller peptides. Peptide is then loaded onto this MHC molecule, and then it is fixed. So now you can see the peptide is there on this MHC molecule. right? And then, yes, it is activating the T helper cells. Okay, so fixation basically means you are seizing all the functions of this particular cell, right? Now in this case you can again see the cell is fixed, so obviously it will not do any activity, right? But we are providing just the peptides from outside, so these peptides can then get loaded onto the MHC molecules. Okay, so you can see here, so in this case, antigen processing did not happen inside the cell. Okay, we are already giving the processed antigen, okay, which is then uploaded on the MHC molecule, and then it activates the T helper cells. Okay, clear? So, is this experimental setup clear to everyone?
Yes, sir.
I will ask this in the next class. Okay. So students, मेक इट क्लियर। सिर्फ एक चीज समझने वाली है, जब हम सेल को फिक्स कर देते हैं तो सेल अपना कोई फंक्शन नहीं कर पाता, एंड इन द फर्स्ट केस यू कैन सी बिकॉज द सेल वाज फिक्स्ड इट वाज नॉट एबल टू डू एंटीजन प्रोसेसिंग, जिसकी वजह से एंटीजन का ब्रेकडाउन हुआ ही नहीं 1 आवर इंक्यूबेशन के बाद भी। ओके, और क्योंकि एंटीजन प्रोसेसिंग एंड प्रेजेंटेशन नहीं हुआ तो टी सेल्स एक्टिवेट नहीं हुए। So this experiment shows the importance of antigen processing and presentation in activation of T cells. Okay?
Yes, sir.
Okay, students, any question at this point?
No, sir.
Okay, students, I think let's have attendance. Okay, by the time you absorb whatever I have explained to you till this point. Okay, so और वो एब्जॉर्ब करते करते अगर कोई क्वेश्चन दिमाग में आए, so you can always ask in that time, and we'll have attendance now, and then we will talk about antigen processing cells, the APCs. What are the different types of APCs? you know we just at the moment know 3 names, but are there any other antigen presenting cells? So we'll see after attendance. Okay, so Yuvraj, Yuvraj, are you there? Yukta,
Present, sir.
Vivek,
Present, sir.
Vanshika,
Present, sir.
Tridev,
Present, sir.
Tanvi,
Present, sir.
Shivani,
Present, sir.
Shakti,
Present, sir.
Sanjana, Sahiba,
Present, sir.
Sahiba, you are not visible. Okay. Rupal, okay, she is present. Nehal,
Present, sir.
okay. Nandini,
Present, sir.
Nandini, you are also not visible.
Visible now.
Namrita,
Present, sir.
okay. Manaswini,
Present, sir.
Kshitij,
Present, sir.
Janvi,
Present, sir.
Ishita, okay. Devanshi,
Present,
Devanshi,
sir.
Sam,
Present, sir.
Present, sir. Present, sir.
yeah, Sam, some people are going behind you. Ayushi,
Present, sir.
Toiba,
Present, sir. Okay.
and Adish.
Present, sir.
Okay. Okay. So Yuvraj, okay, fine. Ishita, you are there. Rupal, you are there. Okay. Okay, students. So let's continue with this processing and presentation part. Okay. Okay, so students, घूम फिर के हम वहीं आ गए इसी पॉइंट पे, राइट? So when it is MHC class 1 molecule, okay, so it will present the peptides, okay, of endogenous antigens, right? And to whom? To कौन से cells? Students, कौन से cells?
Tumor cells. T6 cells.
Yes, and these cells are your CD8 positive T cells. Okay, and if it is MHC class 2 molecule, then it presents the exogenous antigens, right? The peptides of the exogenous antigen to the CD4 positive T helper cells. Okay, so students always keep this in mind: which molecule, which antigen, and to which cell type. Okay, don't make any mistake here, क्योंकि कई बार in confusion you can, what you can do is, एक बार क्या हुआ, I asked, the question was describe the class 1 MHC pathway, right? So student ने class 2 का पूरा pathway सही लिखा था, right? But the question was the class 1 pathway. तो उसको क्या मिला? 0. right? So be very careful कि क्लास 1 पूछा है या क्लास 2 पूछा है, एंडोजेनस एंटीजन पूछा है या एक्सोजेनस एंटीजन पूछा है, CD4 पूछा है या CD8 पूछा है, साइटोटॉक्सिक टी सेल पूछा है या हेल्पर टी सेल पूछा है, राइट? सो इफ you remember this, then obviously you will give the correct answer. Okay, अभी एक दो और terms आएंगे इन्हीं चीज को लेके, right? So be careful with this. Okay, so class 1 MHC molecule, they bind peptide derived from endogenous antigens. So we have just seen that. So if it's a class 1 MHC molecule, so it will present peptides of the endogenous antigen. Right, so I'm not explaining what is endogenous, this you already know, and this their processing we will see in the next class. Then if it is class 2 MHC molecule, then it bind peptide which are derived from the exogenous antigen. Okay, and now this exogenous endogenous antigens should be clear to you because we have discussed this thing twice during the class of antigens and also in the previous class. Okay. Okay. So now before we start with the processing pathway, we must first know what are your antigen presenting cells. Okay, so these antigen presenting cells, they are also called as APCs. Okay, so what are antigen presenting cells? These cells that display peptide associated with MHC class 2 molecules to CD4 positive T helper cells. So antigen presenting cells कौन से होते हैं? Cells that display MHC class 2 molecule on their surface, okay, and present the peptides to CD4 positive T helper cells. तो जो cells ये काम कर सकते हैं, they are your antigen presenting cells. So antigen presenting cell के पास क्या होना चाहिए? एक तो MHC class 2 molecule होना चाहिए, and it should be able to present the peptide to CD4 positive T helper cells. Okay, and the distinguishing feature is that it should have the ability to express class 2 MHC molecule, and it should be able to deliver a co-stimulatory signal. So students, ये सारे points याद कर लो, because suppose if I ask you about antigen presenting cell and you want to get full marks or nearly full marks, right, then you need to write all these things that it should be expressing MHC class 2 molecule on its surface. It should be able to deliver a co-stimulatory signal, okay, and it should present the molecule to CD4 positive T helper cell. So if a cell, okay, is expressing class 2 molecule, can provide a co-stimulatory signal, then it is called as antigen presenting cell. Okay, and now we have professional and non-professional antigen presenting cells. So let's first see what are your professional antigen presenting cells. So these are your dendritic cells, macrophages, and the B cells. And why they are called professional antigen presenting cells? Because they are professional. उनका काम ही यही है. Okay, so ये जो तीन सेल है, डेंड्राइटिक सेल, मैक्रोफेज, बी सेल, इनका काम ही एंटीजन प्रेजेंटेशन का है. ओके, right? So these are your professional antigen presenting cells, क्योंकि इनका काम ही यही है. Okay, apart from these professional antigen presenting cells, there are some non-professional antigen presenting cells. मतलब उनका original काम antigen processing and presentation नहीं है. Okay, but if required, they can act as antigen presenting cells. Okay, So those are your non-professional antigen presenting cells. For example, the fibroblast present in the skin, okay, thymic epithelial cells, thyroid epithelial cells, the glial cells of the brain, okay, pancreatic beta cells, vascular endothelial cells. So they are not professional antigen presenting cell. उनका पहला काम antigen present करना नहीं है, right? But under certain conditions, they can act as antigen presenting cells, okay? So non-professional antigen presenting cells, they does not constitutively express MHC class 2 molecule. Okay, so हमने क्या बोला था पिछली स्लाइड में that antigen presenting cells should have MHC class 2 molecule, right? But these non-professional cells, they do not express MHC class 2 molecule all the time, right? But they express these molecules, as I said, under certain conditions. So expressed only upon stimulation by certain cytokines such as interferon. So students, जब बॉडी के अंदर इन्फेक्शन हो गई, ओके, इन्फेक्शन होगी तो इंटरफेरॉन के लेवल बढ़ेंगे, ठीक है? ये इंटरफेरॉन सिग्नल देगा इन नॉन प्रोफेशनल एंटीजन प्रेजेंटिंग सेल्स को कि भैया अभी इन्फेक्शन हो गई है, तुम सब एक्टिवेट हो जाओ, ठीक है? You act as antigen presenting cells so that we can activate the T cells, so that the immune system पूरी तरह से काम करना शुरू हो जाए, right? So suppose the antigen has entered, in your skin, and they are around your or beneath the skin. So in that case, the fibroblast, they can start acting as antigen presenting cells. Okay, now suppose some infection is there in the brain, and brain may immune system is not as such there, right? So in that case, these glial cells, they will start acting as antigen presenting cells. Okay, so similarly, these are non-professional antigen presenting cells only when the need arise, basically when there is more interferon levels. Okay, so in that case, they will start acting as antigen presenting cells, and that's why they are called non-professional antigen presenting cells. Clear? Any question? Clear?
Yes, sir.
Okay. Now, if we talk about the professional antigen presenting cells, then the most effective antigen presenting cell is your dendritic cell. Okay. And why dendritic cell is the most effective? Because it constitutively expresses high level of MHC molecules and co-stimulatory activity. Okay. तो हमने कहा था जो डेंड्राइटिक सेल है उसके पास ये दो चीजें होनी चाहिए, एक तो MHC क्लास 2 मॉलिक्यूल, ओके। सो डेंड्राइटिक सेल हमेशा ही MHC क्लास 2 मॉलिक्यूल को एक्सप्रेस करके रखता है और हमेशा ही को-स्टिमुलेटरी एक्टिविटी के लिए रेडी होता है, ओके। बट इफ यू टॉक अबाउट मैक्रोफेजेस, so these macrophages must be activated, okay, and only then it express MHC class 2 molecule and the B7 co-stimulatory molecule, okay. तो macrophages के पास ये प्रॉपर्टी है, बट उसको ना थोड़ा धक्का मारना पड़ता है, उसको थोड़ा एक्टिवेट करना पड़ता है, ओके। So it gets activated when it consumes a pathogen, okay. तो जब इसके पेट में खाना जाता है, it gets activated and then it starts expressing MHC class 2 molecule and B7 molecule, okay. So it has to be activated, and then you have the B cells. Now B cells, they are constitutively expressing MHC class 2 molecule, but they express B7 molecule only when it is activated, Okay, so that's why dendritic cells are the most effective, because they are constitutive of expressing both these MHC class 2 molecule and the B7 molecule, whereas others need some activation before they start expressing MHC class 2 molecule and B7 on their surface. Okay, so students, I'll not start with this pathway because this we are going to start in the next class. Okay, so we have already okay, anything that you want to know?
Yes, sir.
Yes, Shakti.
Yes, sir. So you told that proteins are broken down into peptides. So does it happen that some peptides may escape and they do not combine with MSC molecule?
Yes.
Just
Uh,
like potato pieces, they fall from my hand when I ship them.
yes. So I think if you remember, इसमें दो तीन चीजें हैं. One of the thing is, I said that a particular size of the peptide has to be there, right? So if it has to fit into MHC class 2 molecule, it should be around like 10 to 18 amino acid long. अगर इससे बड़ा है, then it will not fit in. एक चीज, ठीक है. एंड हमने शायद जब एंटीजन के बारे में पढ़ा था, सो आई सेड दैट सम ऑफ द पैथोजेंस दे हैव एल एंड डी वाला जो ब्रेक डाउन था, राइट? सो इन दैट केस अगेन सम एंटीजेंस दे कैन एस्केप, राइट? बट अगेन द गुड थिंग इज, जरूरी नहीं है कि एक पैथोजन के सारे पेप्टाइड एक्सप्रेस हों, राइट? एक बैक्टीरिया के या वायरस के selective peptides are good enough to activate the immune system. Okay, basically इस बैक्टीरिया का कोई ऐसा कंपोनेंट जो इसके सरफेस पे है, राइट, इट शुड बी देयर इन दैट पेप्टाइड, इन, राइट? सो बिकॉज जो आगे जो टी सेल्स एक्टिवेट होने हैं, राइट, so some component which is foreign to the host, it need to be presented in those peptides to activate the T cells. Okay, so yes, जो तुम कह रहे हो, बहुत से पेप्टाइड एस्केप होते हैं, राइट? बट अब जैसे तुमने कहा कि कुछ आलू के पीस गिर जाते हैं, ठीक है, बट स्टिल तुम्हारे बाउल में इतने आलू के पीस होते हैं कि यू कैन गो एंड प्रेजेंट इट टू द गेस्ट, राइट? सो यही चीज इसमें भी होगी. ओके,
Okay, thank you, sir.
या Okay, students, I think we can end the class now. दो continuous classes हो चुकी हैं. Lot of information has been passed to you. The only thing is, now you need to absorb everything. In the like previous class, we talked about the MHC molecules. So students, just take out maybe half an hour in the evening या 10 बजे, ठीक है, take a paper, just draw the two MHC molecules. Okay, do that much of work, and then just write down the names of these antigen presenting cells, the professional and the non-professional. Okay, and write down what antigen presenting cells should express. Okay, and do just one more thing, like make or try to understand that, और जो भी तुम्हारा तरीका है उसको लर्न करने का, कि if it's an exogenous antigen, it will use which MHC molecule and it will present to which T cell. ओके, ये तीन चीजें शाम को बैठ के एक बार ओके कर लो. सो आई डोंट थिंक यू विल हैव एनी प्रॉब्लम पूरा आगे इम्यूनोलॉजी समझने में, राइट, बिकॉज दीज आर बेसिक थिंग्स. ठीक है, इसके बाद जब सपोज मैं वैक्सीन पढ़ाने जाऊंगा और उस टाइम पे ये कॉन्सेप्ट की बात करूंगा, तो फिर देन इट विल बिकम डिफिकल्ट फॉर यू. ओके, सो आज ही रात को शाम को ये तीन चीजें कर लो एंड थिंग्स विल बी क्लियर टू यू. स्ट्रक्चर्स हमेशा ड्रॉ करके प्रैक्टिस कर लो, बिकॉज अगेन एंड अगेन आई एम सेइंग lot of students will make that kind of diagram जिसमें बीटा 2 माइक्रोग्लोबुलिन के नीचे भी वो चेन बनाएंगे. ठीक है, बहुत से बच्चे अब भी ऐसी गड़बड़ करेंगे कि अल्फा 1 को बाहर की तरफ ना रख के अंदर की तरफ रख देंगे, राइट. सो जस्ट प्रैक्टिस वंस so that this problem does not happen. Okay.
Okay, sir.
Okay, students, I think then we can end the class, and I think our next class is tomorrow at 12:15. So we will start with one of the pathways. Okay, and on Thursday again we have a class at 9:15. So we will complete both these pathways in this week. Okay.
Okay,
So I'm
sir.
ending the class now. Okay.
Thank you.
Thank you, sir.
Okay. Okay.